Hello, we'll do a quick revision video now on biology paper 1, movement of molecules. So what we're going to be looking at now is we're looking at diffusion, the factors that affect the rate of diffusion, exchange, surface area to volume ratio, and how that's linked to the rate of diffusion. We'll then spend a bit of time looking at osmosis. We'll have a focus on the key practical, which is the effect of a range of concentrations of salt or sugar solutions on the mass of plant tissue, active transport, and then focus on common examples of exchange, and in particular types of specialised cells or specialised surfaces, organ systems for exchange, such as root hair cell, small intestine, and the alveoli. So, type of questions they could give you, very simple to start then, are 3 ways that molecules can move in and out of cells. The only 3 you need to be aware of are diffusion, osmosis, and active transport. So they are the only 3 they will ask you about in GCSE biology. It then could ask you to give specific examples of molecules that move by that type of movement. So diffusion, we tend to mainly talk about gases. So oxygen and carbon dioxide would be the 2 main ones they'll focus about in terms of diffusion. Osmosis is only ever the movement of water. Water only moves by osmosis, and osmosis is only the movement of water. Active transport then, the main examples of active transport are going to be glucose. They might talk about amino acids, although it's highly unlikely. And the most common example I would say that comes up about what uses active transport is the movement of nitrates or mineral ions in plants. You could then talk about identify cells, tissues, organs that are adapted for exchange. So we've spoken about a few of them already. In plants, we're talking particularly about root hair cell. However, we could also talk about palisade cells, which is the site of photosynthesis. In animals then, we're talking more about alveoli. We're talking about a red blood cell, potentially. We're talking about the small intestine. They could talk about, in other animals, something like gills in fish. If in doubt, describe how organisms have become adapted to it. The most common adaptation for exchange is creating a large surface area to volume ratio, or even more simply, a large surface area. The larger the surface area, the greater the rate of exchange. So that would be the best answer. If in doubt, say large surface area. Okay, so let's focus in a bit more detail about the 3 different types. So, in simplified terms, diffusion is the movement from a high concentration to a low concentration. It could be anything. So the most common example, as I said, might be the movement of oxygen from a high concentration outside of the alveoli, inside your alveoli, to then a low concentration inside the blood. Osmosis then is the second type of movement. Any question on osmosis, there are 3 key terms you would need to talk about. So osmosis is the movement of water from a high concentration to a low concentration, but it also has to be through a semi-permeable membrane. And then finally then, active transport Active transport then is the opposite to diffusion. So active transport is the movement of molecules from a low concentration to a high concentration. So what we say for that then is it is against the concentration gradient. And because you're going against the concentration gradient, it requires energy. So just a couple of things to add then. Diffusion is a passive process because it doesn't require energy. Osmosis is a passive process because it doesn't require energy. But active transport, as it says in the name, is an active form of transport because it requires energy. Things to look out for then, if we're talking about energy, that energy comes as a result of respiration, and it comes as a result of there being oxygen, which of course is used in aerobic respiration. So if you see a question talking about the movement of molecules and it shows any form of link to respiration or oxygen or the presence of mitochondria, they are making an assumption that it uses active transport. So just be aware of that. So typical question that might come up in terms of movement of molecules might be something like this. They might have a cell, okay, and then they might do something like this. So imagine these are all substances of some sort. Okay, so I've got three examples here. So what we have then is, you can see here we have one type of movement, here we have another type of movement, and here we have another type of movement. This is water. So it wants you to identify what type of movement is occurring in A, B, and C. So A, here I have a high concentration. Inside my cell, I have a low concentration. So the type of movement is from a high concentration to a low concentration, which means it must be diffusion. B then, it's now moving from a low concentration to a high concentration, which means it must be, for B, where it's going from a low to a high, it must be active transport. C then, the big clue that they've already told you is it's water. I shouldn't even really need to say anything else, but you can also see it's going from a high concentration to a low concentration, which is water. And of course, C then, because it's water, straight away, I know my answer has to be osmosis. In addition to the methods by which molecules move, we also need to be aware about the factors that can affect the rate, which means the speed in which that they move. So the rate is the speed over a given time. So the main factors that will affect it are temperature. If you increase the temperature, you're going to increase the amount of kinetic energy, which means the particles or the molecules or the substances, whichever you want to talk about, they will move faster. So increase in temperature will increase the rate of diffusion. The next one then is surface area. So if there was an area like that, then the number of different entry points or exit points are all of these. Okay. However, if I increase the surface area, so the length is going to be the same, but I've just increased the surface area now, you can see that I have dramatically increased the number of access points where something can get into and out of it. So increasing the surface area has a massive increase in the rate of diffusion. The next one is going to be concentration gradient. Which is the same as saying the difference in concentration. So if you think about diffusion diffusion and osmosis is about the net movement. So if I had a huge concentration here and a tiny concentration here, there is a massive difference between the high concentration and the low concentration. So the probability of things moving from that way to that way are huge. However, if my concentration was like this, the probability of things moving this way is only slightly more than the probability of things moving that way. So if I increase my concentration gradient, the greater the difference in concentration gradient, the greater the difference in concentration, the faster the uptake. The final one then is thickness of membrane, which is exactly the same as the distance for diffusion. The way I like to think about this one is, imagine you yourself are kind of climbing into a garden. If there is one fence to climb over, you are going to be there very, very quickly. Not that I'm encouraging anybody to climb over anybody's gardens. However, if there were lots and lots of fences, it's going to take you a lot longer to get into that garden, which means the greater the distance for diffusion, or the greater the thickness of the membrane, the longer the time it's going to take. So a small, thin membrane means a faster rate of diffusion. A key thing they often talk to you about then is the practical, which is the osmosis practical, which is the effect of a sugar or salt concentration on the mass of plant tissue. So, if we first of all talk then about the title, so it's the effect of salt or sugar concentration on the mass of plant tissue. Okay. So a few handy hints then, first of all. Whenever you see the word effect of, so here, the word that comes after it, or the term that comes after it, is always going to be my independent variable. So the independent variable in this experiment is the salt or sugar concentration. The next clue then is the term that comes after the word on, which in this experiment is the mass of plant tissue, is always going to be my dependent variable. So the dependent variable, which is the one that I'm going to measure, is the mass of plant tissue. Okay, so what does that actually mean? Right, you have a series of test tubes then. And what you might have is a 0% solution, a 50% solution, and a 100% solution. So what that means here is I've got 0% solution, which means it's a lot of water. 50% solution, which means I've got 50% salt, 50% water. And here I've got an 100% salt solution. So what you're going to do is you're going to collect a piece of potato core. Okay, so this is a core piece of potato. You're going to weigh it, and then you're going to add the piece of potato to the solution. You're then going to time, say, 10 minutes. And what you're actually recording is the start mass of the potato and the end mass of the potato, and you're going to calculate the difference in their mass and seeing how it's changed. For the higher tier, to be more statistical and allow you for a valid comparison, you actually want to calculate the percentage change in mass, which is, in order to calculate it, the new mass minus the old mass divided by the old mass, then you times that by 100. Okay? So, a clear thing then for this practical is to talk about the control variables. So the control variables are the variables you have to keep the same. So just a quick recap. Remember, you're changing the concentration of the solution. You're measuring the change in mass. So what are you going to keep the same? Well, you're going to have to keep the same the temperature. Because as we said previously, temperature will affect the rate of movement or the rate of osmosis. So then it's going to affect how much mass the potato gains. You should then keep the same the surface area of potato. Because again, it's one of our factors. So if I had one potato that was that side, and I had another one that was like that, they have a completely different surface area, which is going to affect the rate of uptake. So you have to keep the surface area the same. You should also keep the species or type of potato the same. Okay, so what will actually happen then? And this is the thing that everybody often struggles with. So if I go back to my 3 examples again, 0%, 50%, and 100%. So in the 0% solution, there is more water outside of the potato than there is in it. So water is going to enter the potato, which means 0%, the potato is going to gain mass. At a 50% solution, there is approximately the same concentration of water in the potato as there is in the solution, so it might stay the same. But at an 100% salt or sugar concentration, there is more water inside my potato than there is outside. So water is going to leave the potato, which means the potato is going to lose mass. So what you actually end up seeing is a graph that looks a bit like this. If this graph is concentration of solution on the x-axis and percentage change on the y-axis, the percentage change, you actually see a graph that looks like this. And this value here is very important. This value is where the water concentration in the potato is equal to the water concentration of the solution, which is why there is no net change. Okay, we then focus on examples of cells in particular that are adapted for exchange. So a root hair cell is very well adapted. You can see here then the root hair cell has got a large surface area, which we've spoken about, which means more opportunities to take it up. We also then have here our small intestine. It's got a large surface area. It's also got a lot of mitochondria, which we spoke about earlier. Why does it have mitochondria? Well, mitochondria, sorry about the colour. Mitochondria means it's going to produce energy, which means it can use active transport. So for all of the areas of exchange, we're talking about the same adaptations. So examples, when it talks about examples of exchange, you want them to have a large surface area. So we spoke about more opportunities to get in and out of the cell. You want it to have a very thin membrane. So we spoke about, again, the bigger the membrane, the longer it's going to take to get across it. You want then there to maintain a concentration gradient. And in animals, maintain the concentration is by providing a very good blood supply. So the small intestine and the alveoli, which you can see here, have got what we describe as a copious or an efficient blood supply, which allows always, so every time a molecule moves into the blood, it's taken away. So the blood supply maintains the concentration gradient. Then another adaptation, we just mentioned it a second ago, is for the cells to contain mitochondria. And the final adaptation might be to have ventilation. So something like gills, alveoli, which allows oxygen always to be entering for that exchange. Surface area to volume ratio is a very important one then. Often students struggle to understand what we mean by that. You always have to have the ratio where the volume is at 1. Because what you're saying there effectively, and this is the way I try and explain it, the surface area, if you think about the surface area of the number of doors or the number of entry points into or out of a cell. So what I want to know is how many doors are there per cell. So if there was a surface area to volume ratio of 7:1, the 1 being the volume, this is telling me there are 7 ways in which things can get into and out of my cell. So if my surface area to volume ratio was 0.5:1, it means there's only 0.5 ways to get into the cell, which is not very good. So the higher the surface area to volume ratio, the higher the rate of exchange. You can see there's an example here where they've calculated the surface area to volume ratio. This one would have the fastest rate of exchange because it's got the higher surface area to volume ratio. This type of surface area to volume ratio tends to be reflective of unicellular organisms, uni meaning one. So they can just exchange molecules across their cell surface membrane. As organisms get bigger, which means they're multicellular, it is a lot harder then for them to exchange just across their surface because their surface area to volume ratio is greater. So instead, they have to adapt exchange surfaces. They have to have more specialised ways in which they can do it. So that's why they then have a circulatory system. Okay, these couple of pages here then just talk you through, again, type of questions that are likely to come up and some key information that you need. This talks about the key practical I've mentioned, so it talks about the key words, the variables, a method on the right-hand side, and then what your results table should look like. This is what your describe and explain your answers should look like, and the graph here. Finally then, just a couple of frequently asked questions you're likely to get. So, talks here then, so you can see here, high concentration, low concentration. So name the process in A. I'm going from high to low, which is diffusion. Here, I've got low and high. So when I go from low to high, that's active transport. How are the villi adapted for the efficient exchange? So this is 4 marks, and a lot of you will look at this question and go, oh, that's long, I'm not going to do that. I just very simply go 1, 2, 3, 4. 4 bullet points, 4 marks. It has a large surface area to volume ratio. It is very thin, which means it is 1 cell thick. It also has a very good blood supply. And it has mitochondria, which you can see in the diagram is included. So that's my description. Explain large surface area to volume ratio means there's going to be more area for exchange. 1 cell thick means it has a short distance. A very good blood supply maintains the concentration gradient. And the mitochondria mean I can carry out active transport. This one then is the surface area to volume ratio. So what you'd have to calculate is the surface area to volume ratio of cube X and the surface area to volume ratio of cube Y. So just to recap, to work out the surface area of a cube, it is base times height times 6, because there are 6 sides, and the volume is base times height times width. So for cube Y, it'd be 11 times 11 times 11. And then you have to put all of them in the ratio, surface area to volume ratio, with volume being 1. And the greater the number for the surface area to volume ratio, the better the rate of exchange, which means they're more likely to be. So cube X is my unicellular organism, cube Y is my multicellular. Then finally this looks at the practical. Explain why the potato 0 solution increased. So we've said then, so 2 marks. Why does it increase? It increases because it gains water, because it has the potato has a lower concentration of water. So water enters by osmosis from a high concentration to a low concentration through a semi-permeable membrane. Calculate percentage change in mass divided by the mass. Thank you.